Hi, good morning. Thank you for the introduction, Mustafa, and thank you all of you for being here one more year in Barcelona. So I'm super happy to be with you. Some familiar faces, some new families, and clinicians and researchers. So it's very emotional. So thank you for being here. Okay, this is the title. Okay, My Cells, Bytes, and Chemical Driving Grenopathies, Precision Medicine. So my intention today is to give you an overview of what we do in our group, but in many other groups that are investigating and performing basic and translational neuroscience related to GRIN-related disorders. Okay, it's a bit challenging. I want to show you some background for families and to understand how we proceed when we want to find treatments, which is our common goal. And also I want to present to you an objective view of where we stay right now. So today, the 7th of October, 2023, and what we expect for the next months and years. So having said that, just let me give you another view of what is physiology, what is a human body, how your kids, ourselves, we do function. So this is a very physiological view of what is life. Life we can define at different levels. Levels of energy, levels of chemistry, proteins. We've been talking, we had this beautiful talk from Michelle showing how from a gene there is an mRNA, a protein that assemble, and then they have a function on synapses, okay? So in this scheme, you can find the different dimensions. So starting from basic chemistry, atoms, proteins, there is a structure, which are cells. Neurons are a type of cells. They organize, they constitute tissues, and then they make organs. And at the end, we do have this Vitruvian man or woman. And this is like the most complex that we want to treat. But finding a treatment requires that we go back to the very basics and we understand how genes behave, how proteins interact, how the NMDA receptors or AMPA receptors are being modulated. So we need all this basic knowledge to find treatments and to correct the endpoint, which is the human being. Okay, so this is an animation of the structure, the main actor of our study. This is the NMDA receptor. This is a model that has been developed by Mireia Olivella, where you can find this rotating in this structure. What you can find is this is an NMDA receptor that is acting as an antenna that is collecting all the information from glutamate, glycine, or serine. So this, what you can see here is very interesting, that you see different dots with a color code. So those that are in red are mutations, so point mutations like bricks that are mutated in green patients, okay? And those that are green are positions that are like general variability in the populations that some neurotypical people, they are carrying, but are not pathogenic. So this already shows you that in the NMDA receptors, there are some parts of the receptor, some amino acids, so some pieces of this receptor that are very sensitive. And when there is a mutation affecting this position, it can trigger a disease. But some others, like this one, those that are here, okay, in green, are not vulnerable. So you can affect, you can change, and nothing happens. So the first question that we want to understand is whether one mutation, when you have a green diagnosis, genetic diagnosis, if this mutation is pathogenic or not. So we see the activity of NMDA receptors, and many of you know about what is again the concept of gain or loss of function. But just to give you a very rough view of how they behave. So this is in physiological conditions. The NMDA receptors are like doors that can open on a regulated manner, allowing a calcium influx. So when there is a mutation affecting the NMDA receptors, at the end, we can have like two broad types of mutations that can increase or decrease the activity of these receptors. Biophysically, this is not completely right, okay? So there are intermediate positions, but we need to simplify to find some treatments. This is something that yesterday we had a talk from Dr. Bowes, and we were talking about these complex or intermediate situations. But let's try to simplify. And from this simplified model, the big challenge that we have, and this is one important slide, so please pay attention on this. So once you have the genetic diagnosis, there is a first need that is consisting on the functional evaluation of what is the consequence of a GRIN mutation. So we first want to know whether the GRIN variant is pathogenic or not. This is first important thing. And the second one is to try to put on a box to say, okay, this is a loss of function, or this is a gain of function mutation. This is very important. This will drive the precision medicine. So even the actor is the same, the NMDA receptor is affected, we can have an NMDA receptor that is overactivated, a gain of function, there is too much activity, and we want to compensate, we want to slow down the activity of the mutant receptor. In the other situation, we can have a loss of function mutation, so the NMDA receptor's activity is decreased, and we want to push up, okay? So this is the rationale, and this is what we try to do, we and many other groups. So we move from functional studies to preclinical models. Preclinical models means that we go to the lab and we develop cellular models. We've been, there are cell lines, there are iPS cells, there are also organoids, okay? And also the animal models, zebrafishes, mice models, and so on. But the very important part is also to develop clinical studies, to have a good clinical scale, and to evaluate these potential new treatments that we have defined in preclinical studies. Okay, so let's start. Well, a bit late with the different chapters. So the first one is about bytes, computers. Okay, this is mostly the part of Mireia, so she will correct me if I'm wrong in some aspects. But basically, what we want to do is to take advantage of the massive generation of bioinformatic data, genetic data, the exomes that are being delivered to the scientific community and made available. Also all these omics, this big data science that is also available, and artificial intelligence. So overall, we have like very powerful computational tools that accelerate the research on GRIN-related disorders. So we are using these computational studies to assess the pathogenicity of the GRIN variants of interest, to address the question whether the mutation of my, or the variant of my kid is pathogenic or not, and also to provide this functional stratification. So this was the work mostly of Mireia Olivella and Angel Garcia Cazorla, Adrián Garcia. You are sharing, yes, the first A and Garcia. Sorry, Garcia is a very common name in Spain, so that's the lapsus. Okay, so first thing they created, Adrian and Mireia, they created this GRIN database that is compiling all the genetic information of many groups that have been contributing. Emory's group, led by Dr. Trinellis, Viklik's group as well. So all these publications that have been appeared or databases, we tried to join and to make it available. So this was the project that we completed a couple of years ago. And families, clinicians, it's open and they can check whether the newly diagnosed GRIN variant is reported already or is a new one, okay? So from this database, we found very interesting scientific outcomes. The first one was related to these truncations, to these variants that are promoting a break on the GLUN subunit. So when we talk about truncations, it's a type of mutation that is not introducing an amino acid change, but what it's doing is that it's cutting the protein. So we found something very interesting signature is that mutations affecting the GRIN2A truncations affecting GRIN2A and GRIN2B were associated to a loss of function, while those affecting GRIN1, when there is only one copy, it was not leading to hypofunctionality. So we have been using this database for giving this functional stratification of many variants, but also we have been using this database to complete the structural model and to predict what will be, so to predict computationally, okay, this is the element in the important one, computationally, what will be the consequence of the new missense mutations, those mutations that are not reported yet. So if you take if you pay attention on this model, what you can see is that, that there is, you know, that the NMDA receptors is composed by 4 different subunits. But if you unfold the subunits and you try to put together, okay, it's like putting together my 2 hands. So you have the 2 subunits or 4 subunits, but they are very similar in terms of structure. So the idea was that using the computational tools and validating that these positions are similar, okay, we could extrapolate what is happening for one mutation affecting one subunit to the other mutations affecting another one subunit. So this was the structural superimposition model. Imagine that we do have here on the left, you can see a GRIN1 with neutral non-pathogenic variants that are here in green, but those that are pathogenic are in red. So we have the same, well, we have different variants for GRIN2A and different variants for GRIN2B. Okay, so we extracted, and Mireia extracted all these variants, and she merged all of them. And the final result is that we could theoretically predict what will be the consequence for those mutations that are here and that are not already reported. But we do have the information for this gene. So we moved to the validation. Of course, this was a model, a computational model, but we observed that when we compared the mutations effect, pathogenicity and effects, they were conserved between subunits. So this in the panel A on the left, you can see that there were some coincident pathogenicity or neutrality effects. Sorry, what is that? Yeah, here. And when we compared the certification, those mutations that were loss of function, as in yellow, they were provoking loss of function independently if they were affecting GRIN1, GRIN2A, or GRIN2B. So this was very important because it was accelerating the annotation of new GRIN variants, and this is the final result. So we double mostly the number of disease-associated variants. Those with uncertain pathogenesis were, of course, decreased, and the functionally annotated variants increased by 68%. So that was an important part to increase the number and to provide functional certification of the mutations. But we also, there were still some orphan GRIN variants that were not annotated, neither for GRIN1, nor for GRIN2A, nor for GRIN2B. So that's why we needed to, and different labs are doing the functional studies in cell lines. So basically, this is something that Dr. Viklicki already presented. We used electrophysiological techniques combined to molecular biology. We express the mutant NMDA receptors, and we evaluate. We use this technique to first stratify, so to give a view on what is a loss of, if the mutation causes a loss or a gain of function. But second, this is a very interesting approach also to evaluate the efficacy of new treatments, okay? So I just want to acknowledge that part of this experience of the functional evaluation have been funded by the Spanish Association, GRIN. And then the work was mostly done by Ana Santos and Fede Miguez. Fede Miguez is in the room. So basically, we have all these mutations affecting GRIN 1, 2A, or 2B that have been functionally evaluated in the lab. And this is something that will be kept on with funding of a new generation of students in the lab that are also in the room. So Paula and Gabriel. Okay, so besides this stratification, we really want to develop new approaches and to evaluate new potential compounds. So in collaboration with David Soto, we were trying a new compound that we consider that could be, so we designed and we identified as a potential potentiator of hypofunctional NMDA receptors. So this is a slide, it's pretty old already, so about 4 years ago, but this was a pilot experiment in which we compared the activity. So the 100% is the basal activity of the NMDA receptors, and in gray, you can see the effect of D-serine, okay? And in green is the effect of this new compound that we wanted to evaluate. So first thing is that when we saw these results, we said, okay, we have something, and let's see what happens when we move to a more sophisticated preclinical model. So we moved to the neurons, primary neurons established from mouse embryos, and we observed that these spines, the site of the communication between neurons, were increased when we treated with this potential allosteric, positive allosteric modulator. Then we move to a more sophisticated models, because what happens in a dish is not what happens in nature, real. And we wanted to investigate whether this improvement was also observed in animal models of GRIN-related disorders. So we have several models in the lab, but today I'm going to present you the results we obtained for a GRIN2B loss of function model. So this is a mouse model that has only one copy of GRIN2B, which means that they have less NMDA, less GRIN2B subunits. Okay. This is the model that we're using. Tomorrow, Sylvia will show you different zebrafish models. And we investigated whether this new compound could potentiate ideally this and this could rescue the defects observed in this GRIN2B mouse model. So basically, this is how we proceed in science. It's likely like a clinical trial, but with animal models, you know. So basically, the study design is pretty similar. So we do have the animal models. We compare two groups. This is important. We have a control mice, those that are like neurotypical mice, when they have no genetic alterations. And there is another group, which is this one, the GRIN2B loss of function, okay? They have less GRIN2B expression. And both groups are either treated with the positive allosteric modulator or non-treated. So at the end of this process, we treated chronically with the positive allosteric modulator, and we evaluated whether the alterations of these mice were reversed or not. So how we do? We cannot ask to the mouse if it behaves better, but we do some behavioral tests and some electrophysiological studies. So basically, and I will summarize most of the results, but we observe that the GRIN2B mice, they have normally motor coordination defects and learning and memory defects that are surrounded by brain circuitry alterations. So when we did the test with the different groups, the wild type, the mutant, and then with or without the treatment, what we observed is what you can see here. I'm going to just very quickly show you how we interpret this data. So this is the group. This is we were evaluating how the mouse was performing in a motor coordination test. We put the mouse on a rod. The mouse starts to move and the rod accelerates. So if the mouse has motor defects, it will fall down, not that high, I mean, just 20 centimeters, okay? We follow the ethical rules in the animal facilities, don't be afraid. So the mouse will fall down, and if it has a very good motor performance, it will hold at very high speeds, okay? So this is the measurement on how the mouse has some motor abilities. So observe this graph, please. On the white box, you can see that on average, the wild type mice, they were performing better than the green that are here. Something changed, but okay, on the color, I mean, than the green to be mice, that they were falling down at lower speeds. When we treated this green to be mice chronically with the positive allosteric modulator, we observed that it was an increase, okay? That was significant. Okay, we did the same, exactly the same for the learning and memory task. We present different objects. The mice that have a good memory, they will remind what was the object that they knew before. And what we observed is that the defects present in the green to be mice, they were rescued in the green to be treated mice. And this was grounded by, oops, okay, there is something that is missing here. Okay, sorry. So there is a line here that is related with the green to be mice, and when we treated the mice, we observed that it was an increase of the field potential, so showing that there is an improvement of the brain circuitry, in that case in the hippocampal circuitry. So overall, this evaluation of the treated mice confirm the cellular experiments and show that the treatment with this new compound was improving motor and cognitive skills of the green to be loss of function mice. So I just want to finish. I think I still have 2 minutes. Mustafa, yes? Yeah? Thank you. Okay. So by talking about chemicals, so we are happy with this, with the Elserin achievement a few years ago, and the clinical trial that tomorrow Natalia will show you the good results they obtained. We are happy with this new compound, but still we need to find more treatments. So I think that there is not, let's say, the perfect compound. There are many green genes, green 1, 2A, 2B, 2D, that need a specificity of the treatment. And we only developed for loss of function, but there are also gain of function and complex situations. So the work is not already done, and many labs are investigating new treatments and new drugs that can ameliorate or attenuate the symptoms of green children. So we decided to again to use take advantage of bioinformatics. So we have made an alliance with different pharmas and academic groups to apply for well, a big grant to develop artificial intelligence and to use also structure to define new targets and to evaluate new compounds. So currently, using structural and bioinformatics, we have, Mireia has identified 17 compounds. For some of them, we started the characterization. This is a bit time-consuming project, but we have the advantage of having a good animal models, zebrafish models, that are very convenient for pharmacological screening. And at least from preliminary results, we have observed that there are 4 compounds that are active, so new compounds that are active on NMDA receptors. So let me finish with the translational research. Besides the Elserin trial and related with this new positive allosteric modulator, we have a very initial study on proof of concept clinical study showing how the treatment of 2 patients with this positive allosteric modulator, and this is a representation on the Vineland dimensions before the treatment, so in dotted lines, and you can see how the area is increased after 3 months treatment for both patient 1, but especially for patient 2. You can see that the area is much more increased. So we are already at the end. Let me summarize with this picture of the Sagrada Familia that I hope that you will have time to visit or at least to watch with this city birth tour. So I think that there are the 3 pillars of what we are doing all together. Families and associations, I think that you are crucial. You've seen that not only for funding, but especially for supporting and motivating our groups. You are crucial, and also between you, of course. I have shown you some scientific achievements. So based on the stratification of GRIN variants, new models that, of course, are available for all the scientific community, the proof of concept of this new positive allosteric modulator, and the development of or the evaluation of new compounds. So I think it's also important to remark, and perhaps tomorrow in the roundtable we'll have time for discussing, but I think that there are more and more, this is my optimistic view, no, that there are more research groups, not only in academia, but also pharmas that are being interested, and these are super good news. And we also are contributing to create or to form a new generation of students and researchers that will keep on this research. So I just want to show you the last and the most important slide. So this is, I think, what there is some Irish here. So this is the Rugby World Cup right now, and my favorite team, sorry. But the idea is that I think that we are working together. This is a social event of the lab, and a previous conference that was held here. And I want to acknowledge all the people from the University of Barcelona, Universitat de Vic, Sant Joan de Déu, the clinicians, and the clinics for the contributions. And thank you very much for your attention.